Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss enzyme-linked receptors. In the previous videos, we talked about ligand-gated ion channel receptors as well as G-protein coupled receptors. Today, we'll look at enzyme-linked receptors, and then in a future video, we'll talk about intracellular receptors. Enzyme-linked receptors could either be the intrinsic type or the associated type. So we'll look at the differences between these two. In the intrinsic type, the receptor has a ligand binding domain. So this part here would bind the signal, the ligand, the signal. And at the same time, there is also a domain in the cytoplasmic side of the receptor, which has intrinsic tyrosine, serine, threonine kinase activity. So not only is it a receptor that binds the ligand, the receptor also has enzyme activity, kinase activity. In the associated type, the receptor only has a ligand binding site. It does not have a kinase enzyme activity. But what happens when, we'll see in a few seconds, when the ligand, the signal, binds to the receptor, then it recruits a cytoplasmic kinase enzyme. So that binding of signal to ligand will then recruit an external cytoplasmic enzyme that has kinase activity. Okay, so that's the difference between the two. In this case, the receptor has ligand binding and kinase activity. In this case, it has only ligand binding, but it can recruit a kinase enzyme. Now, why are we quite specific about these amino acids here? Why do we call this tyrosine or serine or threonine kinase? So if you look at the various amino acids that make up proteins, you will see that there are three amino acids that have hydroxyl groups, OH groups, and that is tyrosine, the aromatic amino acid tyrosine, as well as the alcohol amino acids. Serine has a hydroxyl group, and then we have threonine. So tyrosine, serine, and threonine are three amino acids that we find in proteins. So if there's a protein where we see a tyrosine or threonine or serine, that's the only place you see the hydroxyl group. So when we phosphorylate, when we add phosphates to the protein, when the protein is phosphorated, and that's done by enzymes called protein kinases, so you can see phosphates added. The phosphates are always added either on tyrosine or serine or threonine, and once the protein is phosphorylated, the protein is going to change shape, and that change in shape can either increase its function, whatever function the protein has. If it's an enzyme, then the catalytic activity can be increased, or the function can be decreased. So phosphorylation of a protein will affect its function. So this is a very important way of controlling protein functions. We talk about this under regulation of metabolism, but it's just not enzymes that can be modified or regulated by this phosphorylation. Any protein can be phosphorylated. And at the same time, we have enzymes called protein phosphatases that can convert the phosphorylated protein back to the non-phosphorylated form. Okay, so it's kind of interesting that phosphorylation is one of the most important ways to control proteins. And not surprising that phosphate is related to energy, to ATP. Let's look at the enzyme-linked receptors associated with intrinsic kinase activity. First, we'll look at insulin, and then we'll talk about another example, and that's forming growth factor beta. So let's look at the insulin receptor first. We discussed the insulin receptor under metabolism, so here we'll have a quick recap. The insulin receptor is a receptor tyrosine kinase, or RTK. It has 4 subunits, 2 alpha subunits. You can see the 2 alpha subunits, and then you can see the 2 beta subunits. So there are 2 alpha, 2 beta. Now the alpha subunit has the binding site for the signal insulin. So you can see this alpha subunits here bind insulin, while the beta subunits have tyrosine kinase activity. So right here in the cytoplasmic site of the beta subunits, we have the tyrosine kinase activity. So when there is no signal bound to the insulin receptor, basically nothing happens. However, when insulin binds to the alpha subunits, the alpha subunits start changing shape, and because they are connected to the beta subunits, the beta subunits now will start changing shape. And when the beta subunits starts changing their shape, the tyrosine kinase activity of the beta subunits now become active. So now the tyrosine kinase activity of this beta subunit will cause the addition of phosphates on the other beta subunit, while the tyrosine kinase activity of this subunit will cause the addition of phosphate or phosphorylate the other subunit. So each beta subunit catalyzes phosphorylation of about 3 tyrosine residues on the other beta subunit. So now we see phosphate groups attached to the beta subunits on the cytoplasmic side of the receptor. This will now attract a protein called insulin receptor substrate 1, or IRS1. So the SH2 domain of IRS1 will now bind the insulin receptor, and once it's bound, then the insulin receptor, the tyrosine kinase activity of the insulin receptor, will now phosphorylate several tyrosine residues on IRS1. IRS1 is called an adapter protein. Adapter proteins are basically proteins that mediate specific protein-protein interactions. So now IRS1 is bound to the receptor site here. IRS1 is phosphorylated. Remember, phosphates are negative charges. So that means IRS1 now is rich in negative charges, which not only changes its shape, but the negative charges, the several negative charges on IRS1 will also attract proteins which have positive charges. Okay, so this is an electrostatic interaction. So negatively charged proteins due to the phosphates will attract proteins with a net positive charge. That means proteins that has a pi greater than 7 are the ones that will have a net positive charge. So in this way, the phosphorylated and activated IRS1 will now serve as a docking site that will recruit or attract many other proteins, and in this way the message, which is the insulin binding, the message from the insulin receptor due to insulin binding on the plasma membrane, will be then sent to the cytoplasm and nucleus by these various proteins that we'll talk about in a few seconds. And in this way, the insulin signal is transduced into some sort of biological response. So let's look at what happens next. So one of the things that gets attracted to this IRS1 is a protein called GRB2. So when this GRB2 gets attracted to IRS1, that triggers a signaling pathway called the Rus-MAPK pathway. And this is a very important pathway for cell division. So we won't talk about it now. We'll talk about it in a future video. Okay, so that's one of the pathways that will be stimulated by this IRS1 that is interacting with GRB2. Another pathway that is stimulated besides the RAS-MAPK is this phosphoinositide 3 kinase pathway, or PI3K pathway. So let's look at this PI3K pathway. So remember, all of this is happening on the plasma membrane near the receptor. So another protein that is recruited by phosphorylated IRS1 is the enzyme called phosphoinositide 3 kinase, which is the class 1 type. We'll just call that PI3K. So this enzyme PI3K will now be attracted to the plasma membrane where the phosphorylated IRS is. Now, at the plasma membrane, when the enzyme arrives at the plasma membrane, the PI3K will now phosphorylate a membrane lipid that is called phosphatidyl inositol 4,5 bisphosphate, or PIP2. So we have this PIP2 in the membrane, and this enzyme will add a phosphate to PIP2 because this enzyme is a kinase, and the PIP2 will be converted into another membrane lipid called phosphatidyl inositol 3,4,5 triphosphate. So from 2 phosphates, we have 3 phosphates. Okay, so PIP2 will be converted to PIP3 by PI3K. So now we have a lot of PIP3 in this part of the membrane. And this PIP3 will now attract another enzyme called protein kinase B, PKB, which is also commonly referred to as AKT. Okay, so protein kinase B or AKT, same thing. PKB has a domain that is called plextrin homology, or pH, and this was first detected in a platelet protein called plextrin. Like the SH2 domain, this domain, pH2 domain, the pH domain is also found in several signal transduction proteins. Now, another protein that gets attracted to this PIP3 on the membrane is the protein kinase called PDK1. So PDK1 will now bind also at the membrane, and PDK1 will phosphorylate and activate protein kinase B. Okay, so PKB now is phosphorylated, and once it's phosphorylated, PKB now is released from the plasma membrane. It enters the cytoplasm. So the plasma membrane, PKB now enters the cytoplasm. And then PKB goes on to phosphorylate several proteins that we talked about earlier in metabolism. And that is going to cause changes in metabolism, such as increased glucose uptake in muscle cells and adipocytes, increased glycogen synthesis, protein synthesis, as well as lipid synthesis. So this is something we already talked about in a previous video. Let's look at another example of an enzyme-linked receptor associated or that has intrinsic kinase activity. And this is so earlier we talked about receptor tyrosine kinase for insulin. So now we'll talk about receptor serine threonine kinase. And we'll look specifically at this receptor. For the signal here is transforming growth factor beta, or TGF beta. And the receptor is TGF beta receptor, or TGFBR. Now the receptor, this receptor TGFBR, has two types of receptors. There's a type 1 shown in green, and then there is a type 2. So different cells have, there are several different types of type 1 and several different types of type 2 receptors. So it really depends on the cell type, what type do you find, okay, because there are many different types of 1 and 2. So when there's no signal present, here we'll look at the signal TGF-beta, which is basically a protein, a cytokine that is produced by white blood cells. So if there's no signal present, you can see these receptors are separated from one another. Now, once the signal TGF-beta comes and binds the receptor, and it binds at the blue ones here, which is type 2. Once it binds, you can see it brings two type 2 together, and that causes the type 1 to also associate with the complex, and that forms a tetrameric complex. Okay, so two type 2 and two type 1s in the complex with the signal. So this binding now will activate the kinase activity of type 2. Okay, so type 2 not only has a binding site for the signal, it also has a kinase activity site. So this type 2 will now phosphorylate the serine hydroxyl groups on type 1. Okay, and you see that happening, 2 phosphorylating the type 1 receptor. The phosphorylated receptor is now going to recruit proteins called RSMET. RSMET. And RSMET stands for receptor regulated SMET. Now there are several types of RSMETs, including 1, 5, 8, 2, and 3. But the one that is recruited by this TGF-beta signaling is SMET2/3. So this RSMET2/3 is recruited to the receptor, and this is done with the help of this protein called SERA, which stands for SMET anchor for receptor activation. So once this 2/3 comes close to the receptor, now the receptor class 1 will now phosphorylate the SMET2/3 on its serine residues. So once this RSMET residues are phosphorylated by the type 1 receptor, the SMET now is free, it's released from this receptor, it dissociates from the receptor, and now the SMET2/3, which is a regulatory receptor regulated SMET, now it will interact with another SMET, and this SMET is called common mediator SMET or co-SMET. And the only type of co-SMET that we know of today is a SMET4. Okay, so we have a co-SMET that will now interact with this regulatory SMET or the receptor regulated SMET. So once this interaction happens between this RSMET, this receptor regulated SMET, specifically 2/3, with the co-SMET, this is SMET4, this complex will now travel to the nucleus and act as a transcription factor. It will now regulate transcription of several genes, and that will lead to all kinds of changes in cellular activities, such as changes in cell division, differentiation, apoptosis, and so on. It should be noted that besides the RSMETs, the receptor-regulated SMETs, so that's one type of SMET, and then we have co-SMET, the common mediator SMET, co-SMET. They also have another type of SMET called I-SMET, inhibitory SMET. Okay, so these I-SMETs can actually inhibit the R-SMAD activity or the co-SMAD activity. So this is the another example of a receptor that not only has a binding site for the signal, but also has intrinsic kinase activity. There are several other examples of enzyme-linked receptors that have intrinsic kinase activity that we'll talk about in future videos. For example, the EGFR for the signal epidermal growth factor, PDGFR, which is for the signal platelet-derived growth factor, VEGFR for vascular endothelial growth factor, BMPR for bone morphogenetic proteins. Okay, so we will talk about this under at appropriate times in future videos when we talk about cell division or bone formation and so on. Now let's look at the associated type of enzyme-linked receptor, and we'll just look at one example, the leptin receptor that binds the signal leptin. Once again, we have talked about leptin signaling in an earlier video, so just a quick recap. So leptin will bind the receptor, and once the leptin is bound to the receptor, now remember here the receptor only has a binding site for the signal, but it doesn't have a kinase activity. So what happens is once the leptin binds its receptor, now that will attract this protein here. This is a cytoplasmic kinase called Janus kinase 2 or JAK2. So JAK2 gets attracted to this leptin receptor that has a bound signal, and this JAK2 now that has been recruited will now phosphorylate the leptin receptor. So now the receptor is phosphorylated, and once the receptor becomes phosphorylated, that will now recruit or attract several other proteins and will cause the phosphorylation of these proteins. And examples of these proteins that are recruited include STAT, which stands for signal transducer and activator of transcription. Then other proteins that might be recruited include suppressor of cytokine signaling, or SOX, and that will lead to the JAK-STAT pathway. Okay, that will trigger what we call the JAK-STAT pathway. And this pathway will cause the transcription of several genes to be affected. Okay, so these are transcription factors that will go and affect the transcription of several genes. So besides the JAK-STAT pathway, other pathways may also be triggered, such as the MAPK-ERK pathway, as well as the PI3K pathway. Okay, so through this, several transcription of several genes will be affected, and that will lead to various biological outcomes. And some of these biological outcomes we talked about when we discussed leptin under regulation of metabolism in a previous video. Other examples of enzyme-linked receptors that recruit kinases include the growth hormone receptor, the prolactin receptor, the erythropoietin receptor, and several other receptors for signals that include cytokines, interleukins, and interferons. And again, we'll look at these kinds of signaling at appropriate times in future videos. So hopefully this discussion gives you a good idea about enzyme-linked receptors. In the next video, we shall talk about the intracellular receptors. So until the next video, thank you very much for supporting this channel. Have a wonderful, wonderful day. and we'll see you soon.